Greetings, class. Have you ever wondered how a tiny, invisible bubble of oil manages to keep you alive against all odds? Today, we will study the fluid mosaic membrane. In this explainer, we're going to explore the absolutely magnificent architecture that forms the boundary of every single living cell. It's a highly regulated border wall, simultaneously tough enough to protect life's delicate machinery and fluid enough to let it thrive. Let's zoom way in.
All right, let's lay down our foundational blueprint right off the bat. The fluid mosaic model is this brilliant hypothesis proposed by scientists Singer and Nicholson back in 1972. It essentially describes a fluid phospholipid bilayer with a mosaic of proteins embedded right within it. Now, I want you to laser focus on two key words here: fluid and mosaic. Fluid means this isn't some rigid, solid brick wall, right? It's dynamic, it's moving, kind of like a layer of oil floating on a puddle. And mosaic means if you viewed it from above, you wouldn't just see a plain, uniform surface. You'd see this scattered, colorful pattern of proteins moving around, very much like a beautiful tile artwork.
Here are the core details regarding this topic. Review them intently for their being crucial for your finals.
So to really get this, we absolutely have to start with the fundamental building blocks, the phospholipids. Now, these molecules have a bit of a split personality when it comes to water. Because of an uneven electrical charge, the top part, or the head, is polar and hydrophilic, meaning it absolutely loves water. The bottom parts, the tails, well, they are nonpolar and hydrophobic. They are literally terrified of water. So if you were to spread these molecules on a surface of water, they automatically self-assemble into a single layer. The heads plunge right in for a swim, while those phobic tails stick straight up into the air just to stay dry. And this quirky little chemical behavior, it's exactly what gives rise to cells. Okay, so what actually happens when you vigorously shake these molecules up completely submerged in water? Well, those terrified hydrophobic tails are forced to hide. Sometimes they clump together to form spherical balls called micelles, where the water-loving heads face out and the tails tuck safely away inside. But there is a vastly more important formation for us to focus on, and that is the bilayer. Here, two distinct rows of phospholipids align tail to tail, making a perfect molecular sandwich. The tails hide in that water-free center, while the heads face outwards on both sides. These incredibly stable bilayers can fold into three-dimensional bags, forming the absolute foundation of all biological membranes.
Of course, science requires concrete observation, not just neat theories. And zooming in 100,000 times with an electron microscope reveals this exact bilayer architecture in real life. We can actually observe a distinct double black line, which represents those dense hydrophilic heads. Sandwiched right between them is a pale zone where all the tails are hiding. When we measure this entire structure, it averages an unimaginably thin 7 nanometers. I mean, to put that into perspective, you would have to stack about 10,000 of these membranes just to match the thickness of a single sheet of paper. Yet somehow this incredibly fragile barrier is what sustains all complex life.
Let's gather some more evidence by, quite literally, cracking the membrane wide open to find what amounts to a hidden city of proteins. Using a fascinating technique called freeze fracturing, scientists freeze the cell and physically split that delicate bilayer right down its pale, hydrophobic middle. This process completely exposes two internal surfaces. There's the P face, which is the half closest to the cell's inner cytoplasm, and the E face on the outside. Now, looking at the P face under a scanning electron microscope reveals it is heavily studded with bumps. These bumps are actual protein particles embedded deep in the membrane, definitively proving it's a highly complex structure, and definitely not just a plain, empty layer of fat. So bringing all this brilliant evidence together, the micelle chemistry, the double black lines from the microscopes, and those studded proteins we just talked about, Singer and Nicholson formulated the fluid mosaic model. They propose that the membrane has roughly the consistency of olive oil. The individual phospholipid molecules aren't just locked rigidly in place, they're rapidly diffusing and sliding past each other sideways within their own layer. And that mosaic part, that's the diverse, beautiful array of protein molecules floating around in this oily sea. Some are drifting freely, while others are firmly anchored in place. Picture those proteins floating like icebergs in a literal sea of olive oil. But you might be wondering, how does the cell stop that oil from just freezing solid when it gets cold? Well, a massive factor here is the chemical makeup of those hydrophobic tails. Saturated tails are completely straight, meaning they can pack tightly together to make the membrane much more solid. Unsaturated tails, however, possess a distinct kink or bend in them. Because they're bent, they absolutely refuse to pack tightly, they physically push each other apart, loosening up the structure and dramatically increasing the membrane's fluidity. This delicate balance of straight and bent tails along with their length acts as the cell's structural thermostat.
It's almost as if these organisms put on a microscopic winter coat of unsaturated fats to keep their cellular city running smoothly. Look, when environmental temperatures drop, lipids naturally tend to solidify. Just think of a stick of butter sitting in a cold fridge. For simple organisms like bacteria or yeasts that are totally at the mercy of their environment, a frozen membrane is a literal death sentence. So life adapts. They actively synthesize and insert way more of those bent, unsaturated fatty acids into their membranes during a cold snap. By adding those loosely fitting tails, they maintain that critical fluidity even in the freezing cold.
These are the key points related to this topic. Focus on them carefully as they are considered essential for your exams.
Okay, let's look a little closer at the architecture of these intrinsic proteins we've been mentioning. These guys stay firmly locked in the membrane because, well, they hate water almost as much as I hate grading exams. Seriously though, intrinsic proteins embed directly into that 7-nanometer wall. If they span the entire width of it, we specifically call them transmembrane proteins. They stay so well anchored because they contain specific hydrophobic regions, which are very often coiled into alpha helical chains. These helices comfortably snuggle right up against the hydrophobic fatty acid tails inside the core, staying safely away from any surrounding water. You can really think of these intrinsic proteins as floating transport tunnels, or maybe as fixed docks on the edges of our fluid city. While the middle part anchors them down, the top and bottom regions of these proteins are actually hydrophilic, so they happily poke out into the watery environments inside and outside the cell. Sometimes they fold in a way that creates a tunnel lined with water-loving amino acids, making a secure pore that lets water-soluble molecules safely bypass that oily lipid barrier. Now we also have extrinsic or peripheral proteins. These don't embed into the core at all. Instead, they sit entirely on the surface, functioning as vital structural supports or signaling hubs, often binding right onto the intrinsic proteins. Finally, if we're looking at this whole structure from the outside, we see this literal forest of carbohydrate chains adding the final finishing touches to our boundary. Protruding exclusively out into the extracellular space are these tiny, branching carbohydrates. If they're attached to proteins, we call them glycoproteins. If they're attached directly to lipids, they're glycolipids. You can think of them as cellular ID cards, absolutely vital for cells recognizing each other. Furthermore, scattered right in among those lipid tails, you're going to find molecules of cholesterol, which work to fine-tune the membrane stability. All of these incredible components, the phospholipids, the proteins, the carbs, and the cholesterol, coexist dynamically to maintain this unbelievable 7-nanometer biological border. All right, class, let's lock in everything we've just learned with a rapid, high-yield summary of our microscopic architectural tour. Number 1, phospholipids self-assemble into a tiny 7-nanometer bilayer. Number 2, Singer and Nicholson's 1972 fluid mosaic model perfectly confirms this is a dynamic, moving environment. Number 3, bent, unsaturated fatty acid tails are what maintain that crucial fluidity. Number 4, freeze fracturing physically proves the existence of our protein mosaic. Number 5, intrinsic transmembrane proteins act as highly vital transport pores. And number 6, the outer surface acts as a cellular ID forest made of glycoproteins and glycolipids. Make sure you memorize these 6 fundamental pillars of cellular architecture. Before we wrap up, I want you to think really critically about this question as you review your notes today, because truly understanding this is the absolute key to mastering cellular survival. If a simple, single-cell bacteria can adaptively swap out saturated fats for unsaturated ones just to survive a winter frost, consider our own incredibly complex human biology. What exactly would happen on a microscopic level to your cells if they suddenly lost their protective, temperature-controlled environment and couldn't dynamically alter their lipid tails? Would the membrane freeze? Would those vital transport pores just snap under the pressure? Keep that scientific mystery in mind as you study. Thank you for joining me in this explainer, and keep exploring the incredible machinery of life.
Important terminology for this topic is shown on the screen, arranged according to various exam boards. These terms demand focused attention during revision as they are highly significant and must never be overlooked. Furthermore, a past paper evaluation is included for each board and paper format. Give this paper attention too, since it will strongly enhance and refine your preparation. Additionally, aims to grasp the meaning and usage of every term instead of rote learning them individually. Consistent practice of these terms in past paper questions will greatly boost your precision and overall exam results.
Please subscribe our channel to get more educational videos. Thank you.